Hello everyone, welcome back to our channel. This is Dr.Nazma and in this video we are going to continue the NCERT Solutions of the chapter Some Basic Concepts of Chemistry. So let's get started with the very first question of this video, 1.16. So in fact the remaining questions like the questions 1.15 and below have been covered in the rest of the parts like part 1, part 2 and part 3. You can avail the links in the description of this video if you want to watch those questions and the solutions. So let's get started. What do you mean by significant figures? This is totally a theory-based question. Here is the answer for this question. Significant figures are those meaningful digits that are known with certainty, and they indicate uncertainty in an experiment or calculated value. Say for example, you're talking about a figure 15.6. This is measuring the volume in ml. The result of the experiment 15.6 is saying that 15 is a certain value and 0.6, this 0.6 is an uncertain value. And the total number of significance figures in this particular number are 3, 1, 5 and 6. And significant figures are defined as the total number of digits in a number including the last digit that represents the uncertainty of the result. So knowing the definition is not so very important, but being able to identify the significant figures is important. We'll be solving one such question going ahead. Now quickly moving on to the next question, 1.17. A sample of drinking water was found to be severely contaminated with the chloroform, which is CHCl3. And this is supposed to be carcinogenic by nature. This chloroform is carcinogenic by nature. The level of concentration or level of contamination was around 15 parts per million, which is ppm by mass. So now if this is the description of the question, you got to express this 15 ppm value in terms of mass percent or percent by mass. Similarly, determine the molarity, which is small m, of chloroform in water sample. So now you have been given a scenario in which the concentration of your chloroform in water is 15 ppm. Convert this into percentage mass first. Let's see how to do this question. So we are talking about 1 ppm. What is meant by 1 ppm? 1 ppm is 1 parts per million, saying that 1 part of the solute is present in 10 power 6, which is 1 million parts of the total solution. Say if I'm saying that the concentration of a solution is 1 ppm, maybe 1 ppm chloroform solution. It means I have 1 part of chloroform in 10 to the power 6 parts of the whole solution. Fine. So similarly here they are saying that it is 15 ppm of chloroform. So this 15 ppm of chloroform in water means you are having 15 parts of chloroform in total 10 to the power 6 parts of water. Fine. And now when you try to convert this into mass percentage, you simply have to multiply this with 100. Why? Because in the question they already have said that you are talking about ppm, parts per million, in terms of mass. That means you are saying that 15 grams of chloroform is present in 10 power 6 parts of water. So you are already talking in terms of mass. So 15 divided by 10 power 6 is directly multiplied with 100, which on simplification is 1.5 into 10 power minus 3 percentage. This is the mass percent of chloroform in the given sample of water. And now the second part of the question is determine molarity. We know that molarity is concentration with reference to the mass of solvent in the given solution. So let's first understand what do we have. We have 100 grams of the sample, which is consisting of 1.5 into 10 power minus 3 grams of CHCl3. And now if you ask me, ma'am, what is this 100 grams? Because you're calculating in terms of percentage, right? So this 1.5 into 10 power minus 3 is per 100. It's per cent. Cent is 100. So in 100 grams of water sample, you have 1.5 into 10 power minus 3 grams of CHCl3. So you know the mass of CHCl3. And now in case you're talking about 1000 grams, then what does it mean? If you're talking about 1000 grams, it means in 1000 grams, you will be having 1.5 into 10 power minus 2 grams of CHCl3. And how is this possible? Through this cross multiplication. If 100 grams is having 1.5 into 10 power minus 3 grams of chloroform, 1000 grams will have how much? 1000 into 1.5 into 10 power minus 3 divided by 100, which in turn gives you 1.5 into 10 power minus 2 grams. So now this is the mass of chloroform. We know that molarity is equal to number of moles divided by the weight of the solvent in terms of kilograms, correct? But now what do we have? We do have 1000 grams of sample consisting of so and so grams of chloroform, which means you already have considered W in your formula. You already have considered mass of solute. So N is equal to weight by gram molecular mass of solute, right? So you have considered W, you already have considered small w. So now what is left out is molar mass. Molar mass of chloroform is 119.5. Now substituting all the values that you know, substituting 1.5 into 10 power minus 2 divided by 119.5 into 1 by the total mass of the solvent, which is 1000 grams. Substituting all of this and simplifying, you will be having 1.25 into 10 power minus 4 molality. Molality is represented by small m. I hope you have understood this. But then one link that you need to remember for sure is of molality. Molality is weight by gram molecular weight of solute into 1 by capital W. All the known values are being substituted into this molality formula. Hoping that you have understood this. Moving ahead with next question, number 1.18. Express the following in scientific notation. So you're having a few terms over here, a few numbers here. You got to express them in scientific notation. I'm sure you know what is scientific notation. Scientific notation is you converting the given number in terms of multiples of 10. Maybe 10 power plus 1 plus 2 plus 3 goes on, or maybe minus 1 minus 2 minus 3 goes on. So now say for example you're talking about 0.0048. Now in order to convert this into scientific notation, what am I doing is I'm moving this decimal, this point towards my right hand side. So when we move it towards right hand side, when you want to convert this into scientific notation, over 10 in the power of 10, you will be increasing the minus value. Say for example, point moved by 1 decimal. Point moved by 1 decimal is 10 power minus 1. Point moved by the second decimal, 10 power minus 2. Moved by third decimal, 10 power minus 3. As you gradually move towards your right hand side, in the power of 10, the minus value goes on increasing. One place movement is equal to minus 1. So in that regard, the number that we have right now is what? 0.0048. It's being converted into scientific notation. Moving the point by 1, moving the point by 2, moving the point by 3. So it has to be 10 power minus 3. 4.8 into 10 power minus 3. If you still want to convert it, you can also write it as 48 into 10 power minus 4. Okay. Next one, 23. Okay, it is 234,000 being converted into scientific notation. So since I don't have a point over here, I try to put a point. If I place a point over here, and if I place a point over here, if I place a point over here, if we place a point over here, so gradually what am I doing? I'm moving towards my left hand side, which means moving towards left hand side means in the power of the 10 you will be increasing the values by plus 1. So say for example in 234,000, initially I have moved the point by 1 decimal, so it's got to be 10 to the power plus 1. Moved by 2 decimals, 10 to the power plus 2. 3 decimals, 10 to the power plus 3. 4 decimals, 10 to the power plus 4. 5 decimals, 10 to the power plus 5. So it's finally 2.34000. You don't need to represent it though, because you're representing it here. 10 to the power plus 5. Plus does not need to be represented, so I'm writing it as 10 to the power 5. I hope you're understanding. Now if you are understanding this, you try this by your own. But then in case you're stuck, listen to me. Cool. So you're having 8008. I want to convert this. The main intention of this scientific notation is to be able to write everything within the numbers 1 to 10. Okay. So now I want to write it within the numbers 1 to 10. So I'll imagine a point over here and then a point over here. I'm sorry for this. A point over here and then a point over here and then a point over here. So gradually I'm moving towards my left hand side. So in the power of 10, I'll be having plus value increasing. How many points have I moved? 1, 2, 3. So it's got to be 8.008 into 10 power 3. I hope you got this correct. And now the next one is 500.0. So what am I doing is I'm moving it and moving it by two symbols or two positions. It will be 5.000 into 10 to the power 2. You don't need to represent this 000, but just showing in order to avoid your confusion. Now 6.0012. It's already written 1 to 10, the range of 1 to 10, right? You don't need to convert it into any more significant or scientific notations. It's already in there, right? So scientific notations, the main intention is to be able to convert them into the range of 1 to 10. So if you're moving the point towards your right hand side, the minus value increases. If you're moving the point towards your left hand side, the plus value in the power of 10 increases. Now moving on to next question. Number 1.19. How many significant figures are present in the following? You have 6 values here. You got to find out how many significant figures are there in these values. So I'm sure you have learned the rules to identify significant figures. If you have not, please do that, because identifying significant figures is an important question from this chapter. So the first one, 0.0025. So in the first one, how many significant figures do I have? I just have 2 significant figures over here. The 0 which is present just before the point and zeros present after the point are non-significant. Second figure, 208. If you have a 0 suspended between two other numbers, the 0 is also significant. So including that 0, I have 3 significant figures, 2, 0 and 8. Third one, 5005. The same rule applies. 0 suspended between two figures has significant value. So you call it as 1, 2, 3, 4. 4 significant figures in total. And here, the zeros which are present after the comma are non-significant. So I have 1, 2, 6. 3 significant figures only. And now the zeros which are present over here between the point and the number are significant, and the 0 which is immediately present after the point is also significant. So 1, 2, 3, 4. And now finally in the sixth one, you are having totally 5 significant figures due to the same rules. The 0 present just immediately after the point is significant. Cool. So here you go with the significant figures and identifying them. Next question, number 1.20. Round up the value up to 3 significant figures. You got to round up these values in such a way that you get 3 significant figures remaining. Cool. So now in the first case, I'm rounding up the value 34.216 in such a way that I get 3 significant figures, 34.2. So now the second case, I'm having 10.4107. So now I'm rounding up this value in such a way that I have only 3 significant figures. So I'm avoiding this 10.4. In the third case, I'm rounding up in such a way that I get 3 significant figures. But if you look at the answers, 460 is what you have. Why? In order to have 3 significant figures, you could cut down this, right? But then after cutting down this, you are having 4, 5, 9, right? So this can be further rounded up to 460. That's what is here. And now the next one is 2808. In order to get a significant value, 3 significant figures, that 08 is rounded up to 10. 2810 is the resultant answer. So this is how you identify significant figures. I hope you are understanding all the solutions, but in case you are not understanding them, please make sure you ask me your doubts in the comment section below. Hoping that these videos are helping you. I'm signing off for today. So I'll see you in one other video. Until then, bye-bye everyone.